pleasure to present to you Professor Robijn Bruinsma. He's a theoretical physicist, professor of physics at the University of California at LA, and chair of the Department of Theoretical Physics for the Life Sciences at Leiden University. He got his PhD in physics from the University of Southern California. Since 2001, he's a fellow of the American Physical Society. In 1999, he was a distinguished lecturer at the College of France. In 1996, he got a Rothschild Foundation Fellowship. And in 1994, he got a Pierre et Marie Curie visiting professorship. He's a specialist in condensed matter theory, and he has a lot of interest, but now his interests are related to numerical simulation of active proteins and gene transcription, self-assembly of viruses, self-assembly of DNA, and the physics of chromatin, electrostatic of DNA, and electrical transport along DNA, adhesion of vesicles and cells. So, Robijn, welcome to our colloquium. Thank you very much.
Orlando, thank you very much for your introduction. I need to start with an apology that I cannot be with you. I have visited your beautiful city a number of times, and I was looking forward to visit UNAM. I have visited UAM, your sister university, many times, and unfortunately, I wasn't able to do it this time because of the epidemic. I also apologize for not speaking Spanish. I am trying, but it is pitiable, so I will not expose you because you might die of laughter. The third reason I need to apologize is because if I would go to a talk with the title Physics and the HIV virus, I might expect reasonably that physicists, that people would expect physics to contribute to therapy. And nothing I shall say today will have anything to do with therapy. In fact, it is physics. And the reason why I'm talking to you is because HIV is maybe the most well-studied virus. It has an enormous number of articles dedicated to it, and we know a great deal about it. And because we have learned a great deal about it, it became clear to me and co-workers that there are mechanisms at work which we cannot understand with our immediate physical intuition. So I'm not talking here about problems involving therapy. I'm talking here about how it works. And how it works seems strange for multiple reasons. Now, what I will talk to you about today are just two cases, and I've called them the adventure of the stalled butt and the adventure of the second shell. And the choice of those two words, those two titles, comes from the stories of Sherlock Holmes, which you see over here, a picture, because what we are doing in this context is much more like Sherlock Holmes than the type of physics you're familiar with. I am by training a theoretical physicist, and I love theoretical physics. You start with a set of fundamental principles like Maxwell's laws or something else, and then you construct, using mathematics, beautiful descriptions which you then test against nature. That's the heart of physics. It doesn't quite work like that if you work on something like HIV. Instead, you get bits and pieces of information which may or may not be relevant. You have to put them together and see if with the tools of physics, you can do something useful. So Sherlock Holmes gave me a sketch of the events. Here you see Sherlock Holmes and Dr. Watson in a train, and they sketch events. And that's what I'm going to try to do today for you. Now, I am very used to interruptions, and I actually need your interruptions. And so please interrupt me. I don't know if the chair can give you how you want to do that with raising hands, or with, I cannot see your chat, but if you can simply indicate to the chair that you'd like to ask the question, I would really ask you to so I can explain it to you, so you can benefit from the talk. So I'm going to start with a picture of HIV. And our standard way of making pictures of HIV is cryo-electron microscopy. The characteristic size of an HIV virus is about 100 nanometer, 1000 angstrom, which puts it below the limit for conventional optical microscopes. So for a long time, cryo-electron microscopy has been our standby. So here you see an old picture, but one which is still very often shown, of an infected host cell. So this is the length scale. The infected host cell is here, where you see this dark. Outside here is fluid. And this thick line you see over here is a normal part of the cell. It's called the plasma membrane. All sorts of stuff is transported through the plasma membrane, including water, but also ions and nutrients. So it is this membrane, which will play a big role in my talk, which keeps the inside of a cell from the exterior. Now, the sign of the infection here of the cell is this thick layer you see over here. And it's composed of proteins, and they're called capsid proteins. So these proteins will spontaneously assemble under the right circumstances to form a closed shell, which is called the capsid. That hasn't happened here yet. So at this point, they only have kind of like a half shell. So in terms of length scale, these are enormous proteins. A typical protein is about a nanometer. So that is 1 on this length scale. These proteins are in the range, this length scale is in a range of 10 nanometer. So about 10 times bigger or more than an ordinary protein. So this is one of the key components of the growing virus. So the other key component is the genetic molecule, which in this case is RNA. I'll tell you later about the difference between RNA and DNA. So here you see the beginning of the development of a new virus. The RNA is adhering to the inside of the shell of proteins. So we call this a bud, of course, after flowers, budding flowers. Of course, this is a very nasty bud. It's a viral bud. So I'm now going to show you a picture which is at the next stage of the development of the virus. At the next stage, the shell has closed. You can see it. But look carefully. This thick shell is interrupted. It is broken over here. You can see there is a reduced density. So the protein, this thick protein layer, which in one part of its function provides mechanical protection for the RNA, here is not complete. There's a hole there, and we'll see later there are other holes. That's quite strange for a person like me who works on viruses, because the standard virus I'm familiar with is beautiful icosahedral shells with perfectly ordered proteins. That is evidently not the case here. These capsid proteins are not positionally ordered. You can make higher resolution images and you can see that it is quite defected. And this hole already speaks for itself. You can also see that the lipid membrane, the membrane of the cell, the plasma membranes have been interrupted and broken. This might be some content of the cell which is leaking out at that point. So there is the hole. And we have a single thick shell at this point. You call this stage the immature virus, and it is not infectious. These might be proteins which are embedded in the membrane. So the cell membrane of the host continues to cover it. So it has stolen a closed shell of membrane with some additional proteins which the virus synthesized inside the host cell and which are now placed on the surface. But this structure is not infectious. It cannot make you ill. Then, very unusually, this shell evolves. It is a little bit as if it is alive. Normally, this doesn't happen. Normally, viruses are inert. This virus evolves once, even after it is outside the host cell. It spontaneously evolves from this thick shell structure to two thin shells. So you see here, you can actually see the plasma membrane a little bit better. You lose all of this heavy density, and there's a much lighter density left. That's the outer shell. And then you see here this cone-like structure. Here there are two of these images. And that's like an inner shell. The RNA, the viral RNA, which originally adhered to the outer shell and still does so over here, has been released, and it is now captured inside this inner shell. So the first thing you would think about from an engineering viewpoint is, why do you need two shells? The reason why you need a shell for any virus is because you need to protect the RNA from digestive proteins and from mechanical stress. So that's the standard role of the viral capsid. Here we have two. We have an outer capsid and an inner capsid, and that is strange. This is a key feature which you see of a class of viruses called the retroviruses. The HIV virus is a retrovirus, which makes it fundamentally different from the polio virus, the common cold virus, or even the coronavirus. So the second title of the riddle of the second shell is here. Why are there two thin shells? And when you talk to a molecular biologist, they generally don't understand what you're talking about. They will say, well, that's the way the system evolves. For a physicist, that's not a satisfactory answer. There must be a clear functional reason why we have a second shell here. And it must be, this must be something which is increasing the viability, the ability of the virus to, through the host cell, reproduce itself. I want to emphasize that viruses are parasites. They can't reproduce by themselves. They always need a host shell machinery. Before I go on, are there any questions you would like me to address, or is things are clear? So please let me know if I'm going too fast or too slow. So now I'm going to show you something you might see in a textbook. So this is a cartoon where you simplify what you just saw. Here is the membrane of the cell. So this yellow line. Here is this thick layer of capsid proteins we saw in the first part. So this is the budding, this is the assembly stage. This very thin red line, that's the RNA, which is sticking to the inside of this shell. So this budding completes, the hole here is indicated. Then you have a very narrow neck, which disappears, and the immature virus forms through this budding and pinch-off process. Then this strange, massive transformation where the complete structure changes is called maturation. The mature infectious virus has an outer shell, here in yellow, with the cell membrane still there and a fragment of the proteins, and then an inner shell. And this inner shell contains the RNA molecule, and it is not adhering to the outer shell. So I need to immediately tell you what is happening during maturation. And that is also something a physicist is startled by to learn. The protein, this large protein, at this stage, during maturation, gets cut up. It cuts, in fact, it cuts itself. It cuts itself at 5 different places. It's only this top part, this red thing, which remains here. So this red color here corresponds to this red color. That's a fragment of the initial protein. This part of the protein forms the green shell of the mature virus, the second shell, and I'll later tell you about this part. So, I need to tell you also a little bit about the nature of this yellow sheet, which surrounds both the cell and which envelops the virus. So that's a cell membrane, and it's a lipid bilayer, and it's a 2D fluid. So with a lipid bilayer, I mean that this is a group which is charged or dipolar, and it is hydrophilic, it is water-loving, and this is a tail group, which is aliphatic, which is apolar, and which doesn't dissolve in water. So the net result is that if you dissolve these type of lipid molecules, of which you have a great deal, in water, they spontaneously form bilayers. We call that self-assembly. And Rolando can tell you, or has told you, I'm sure, a lot about lipid bilayers, which he's an expert. So we have here a 2D fluid. It's flexible. These molecules move around, and they, because it is fluid, allow shape deformations to happen. So again, that'll take an important role in my talk. So we use the word hydrophilic for the polar head groups, hydrophobic for the apolar tail, and so they spontaneously assemble. So now I can start my first adventure after this introduction. So what you see here is a picture which is of higher quality, it's more recent, and the authors of this picture were able to image the whole structure of the immature capsid. So this is the immature capsid, and I already mentioned there are holes there. This is the big hole we talked about before, and these are smaller holes. So the budding process is completely spontaneous. It doesn't consume molecules like sugar or high energy molecules. It's a spontaneous thermodynamic process which lowers the free energy of the system. It is spontaneous up to a point. It stalls. Just at this stage, just before the virus is going to leave the infected host cell, it stalls and it waits. And that is strange. We'll come to that in a second. The actual pinch off, the release of it, takes place after the virus, or rather after the viral proteins have recruited host cell proteins. So something happens, and we know a great deal about it, to cause a certain group of cell proteins to come to the neck and to form a ring. These are active motor proteins, and they can contract, much like a muscle contraction, and there cause the pinch off to happen. It's complicated. It's called escort machinery, and it's used a lot in cells for a normal process called endocytosis. So it's a natural function, and typically of a virus that gets hijacked to pinch off the virus just after budding. Now, again, from the viewpoint of a life scientist, that would seem very nice, and you should study the escort machinery. From the viewpoint of a physicist like me, it is very unsatisfactory, not because I doubt this, the evidence is overwhelming, but why? And why do I mean with why? So lipid membranes have been studied now for decades. And one of the results of these studies of lipid membranes is that lipid membranes can produce very similar looking lipid bubbles without stalling. It is perfectly spontaneous. So why does the HIV virus even need the escort machinery? Why doesn't it go spontaneously? So the theory for this budding of lipid membranes was written down by Reinhard Liposomes, a German theoretical physicist, and it was done in 1992, more than 25 years ago. But it is still very pretty, and I want to show you how it works. So this does not happen if you have one kind of lipid, but it does happen if you have two kinds of lipid, alpha and beta. And in the Lipovsky model, these two there, by the way, we have like 35 different kind of lipids in our cell membranes. These two lipids do not mix, okay? And so you get phase separation, but this is not two-dimensional phase separation in 2 dimensions, which is perfectly ordinary. It is something hybrid. It's a two-dimensional phase separation in 3-dimensional space. So let me show. So this is initially a uniform sheet, which is a uniform mixture of lipid alpha and lipid beta. Time t equals 0, you let it go. Alpha and beta do not mix, and let's say beta is the minority component and forms 2-dimensional droplets where it condenses out. So far, so boring. That's a standard phenomenon. But as for any type of phase separation, there is line tension. Well, typically it's surface tension in 3 dimensions, but here it's line tension. Now imagine that you have a very deformable flat disk and you want to contract it. This interfacial line tension wants to squeeze it. Now, if this was in a 2-dimensional space, nothing would happen. It just compresses it. You call it capillary pressure, and that would be that. However, because it is embedded in 3-dimensional space, it's going to pop out into the 3rd dimension. It can reduce the length of the interface by deforming into the 3rd dimension, and then it perfectly spontaneously pinches off. You don't need to do anything. So these lipid buds pop out into the 3rd dimension entirely due to line tension. There's no stalling, and it's a perfectly natural process. So since lipid bilayers are available, why doesn't HIV use this perfectly workable mechanism? Okay.
Excuse me.
Yes, please.
Is there any Gaussian curvature term involved in this process?
I'll come to that. That is the heart of my talk.
Oh, shit.
So
sorry.
I'm going to come immediately to Gauss curvature in a moment. So for those of you who know about lipid bilayers, normally we explain this in terms of what's called the mean curvature, which is what you ordinarily assume with curvature. So you have a competition between the curvature energy cost of this bubble and this line tension, which tries to squeeze it. So let me go to that. So I am a theoretical physicist and I cannot resist equations. So there is a beautiful continuum theory of symmetric liquid surfaces. So I'll tell you in a moment what I mean here with symmetric. So here you have a surface, a liquid surface, and its height profile is determined by the function f of r. So this is the location r, and this is the height of the liquid surface above r. Okay. With symmetry, I mean here that the system is supposed to be translational invariant, rotational invariant, and up-down symmetry. A lipid bilayer has up-down symmetry. If you reflect it along this midplane, it stays the same. Of course, you can have all kinds of molecular surfaces which lack that. So let's try to write down something like a Landau energy for such a liquid surface and how it bends, obviously inspired by the budding question. Well, you characterize the shape of such a surface by the curvature tensor, which is just the matrix of second derivatives of the height. So it's a 2 by 2 matrix because you have 2 coordinates, x and y. So you have 2 by 2 matrix, which completely characterizes the curvature of this surface. And in the spirit of Landau, we now need to construct our energy as invariants under the symmetry operations, these 3 symmetry operations: translation, rotation, and up-down symmetry. And Landau instructs you to find the lowest order invariants. And the lowest order invariants are related to the trace of the curvature tensor and the determinant of the curvature tensor. And the 2 lowest order invariants are the square of the mean curvature and the Gauss curvature itself. So the question you just heard, how about this Gauss curvature? Does it play a role? Let's address it right away. So we're now going to construct a simple free energy from the mean curvature h and the Gauss curvature k. And so this curvature energy is some functional of the height profile, and we're going to construct it from the lowest order invariance of the curvature tensor. Here it is. Here are the two terms, h squared, the trace of the tensor squared, and the determinant. So this has dimensions of 1 over length squared. You integrate it over the surface, so the proportionality constants have units of energy. There are two. Then the first one is called the Helfrich bending modulus, and the surface is unstable if it's negative, so it has to be positive. And the second one is the Gauss bending modulus, which for lipid bilayers is negative. So both have the units of energy, and for lipid bilayers, it's about 20 kT. And kappa bar is typically comparable to kappa. So why would you call this even the Gauss curvature, this determinant? That's because of a theorem from mathematics. And a theorem from mathematics says that if you treat this as a constant, you can bring it out, then the remaining integral of the determinant of the curvature tensor is topological invariant. It's the same. You can change the shape for a closed surface. Any way you want it, this number will stay the same. For a simple closed surface without handles, it's 4 pi. So that's it. So typically when physicists study lipid bilayers, we ignore this term because it's not going to do anything. It's just fixed. You can vary things. If you minimize it, it stays what it stays, but it is what it is. So that's boring for most cases. But Liposomes already noticed that this argument doesn't work if you have this mixture. Because you have this mixture, there is an interface here between the two types of lipids. And kappa bar could very well be different in the beta and in the alpha phase. And then this is no longer equal to 4 pi. And he included that in his description. I wish I could tell you, but we don't have time for it. So we have a pretty complete theory. This theory, which is called the Helfrich-Canham theory, and it has been tested for 30 years. And again, Rolando will be able to tell you a lot about it. The key point is that in this theory, there is no stalling. And if you do experiments on the budding of lipid vesicles from lipid bilayers in this manner, there's no stalling. So that is strange. So if you mention that to a molecular biologist, you say, come on, what are you worried about? This is an incredibly complicated system. There are a million proteins around. I don't know. What would he wouldn't even, he would just kick you out of the door because the example of the Liposomes model is so trivial that he wouldn't even be concerned, or she, excuse me. However, and that may very well be true. We looked at the escort machine. It's very complicated. But there is something here which could make you wonder whether the Liposomes theory applies. And the reason is the proteins. The proteins for viruses which are enveloped, the capsid proteins are always asymmetric. They break the up-down symmetry. In fact, they are curvature generating proteins. If you have a cluster, so this is nothing specific for HIV. This is a transmembrane protein, and I should hasten to say that the protein which I showed you earlier for HIV is not a transmembrane protein. It doesn't matter. These kind of proteins, when they cluster together, curve the membrane. So that's the reason why it starts to curve. When these capsid proteins come together, they're generated by the viral genome, the proteins, they go sit in the membrane. And when they come together, they spontaneously curve the membrane and it breaks the up-down symmetry. So from the viewpoint of Landau theory, you cannot really use, for this case, the Liposomes theory. So what do? We use simulations. So I am really ignorant on simulations. And this was not done by me, but by a collaborator who was doing beautiful numerical simulations, Michael Hagen from Brandeis University. And here you can find the reference. So this is a numerical simulation of the budding induced by capsid proteins. Incidentally, you could very well treat this as a form of phase separation. And maybe it's the line tension of this aggregate with the with the lipid membrane, which is causing the budding. That's entirely possible. So we're going to make a toy model to test this. And the toy model starts with a toy model for the capsid protein, and that is this rigid cluster. So we take here a rigid cluster of these 3 purple spheres. These 4 times 3 is 12 green spheres, and then 3 purple spheres from the bottom. It looks sort of like 3 ice cream cones which I stick together. And the rules for our toy model is purple repels purple, purple repels green, and green attacks green. That looks arbitrary and pointless. At this point, we just want to think about the effect of curvature generating proteins in a lipid bilayer. So because green attacks green, if you take 2 of these proteins, they will want to stick together by sharing an interface. And the energy, if you want to estimate it, to make it reasonable, it's just a few kT. So what's going to happen if you have a whole cluster? Then what's the minimum free energy state? Well, it is a hollow shell. And you form these, and they just grow until they close on themselves. And the radius of this hollow shell is just set by the angle. So you can construct an angle here by extending that, and that angle will be roughly the center of this shell. So the cone angle, for the system to be in mechanical equilibrium, the cone angle basically sets the size of the preferred shell. So now we have a mechanism to form capsids with these toy proteins. Go on, and we go on with model lipids. So we're going to do the same thing, except our model lipid is flexible. It consists of 1 blue sphere and 2 yellow spheres. And we play the same game. Blue repels blue, blue repels yellow, and yellow attracts yellow. If you take tens of thousands of these molecules and you do a Monte Carlo simulation or a molecular dynamic simulation, you get formation of this kind of structures. Because yellow attracts yellow, you want to, you'll get an aggregation. But that would still leave yellow exposed to the fluid environment, and you can gain additional lower additional free energy by making a bilayer. So you get in the computer simulation spontaneous formation of lipid bilayers. You are hiding the yellow hydrophobic part. So this is like the aliphatic tails, and this is like the polar head groups. So this blue is the same as that green. I apologize. And if you make the attractive energy sufficiently weak, this thing will be thermally fluctuating. You will see that there are some of these molecules which escape, they evaporate, and they later might come back. And it's kind of wobbly and wiggling. I'll show you a movie pretty soon. So what you then do is you insert the model proteins in the model lipid bilayer. So here you do it. I on purpose show here the original lipid bilayer cartoon, and now we insert it. And the way we do that is by simply making the final rule green attracts yellow. Now, I want to ask if there are questions about this model. So this is a really coarse-grained toy model. It by no means represents HIV. Is the model clear to everybody? Or would you like me to explain it a little bit more?
In these molecular calculations, do you have water or not?
No, there's no water.
Okay.
You keep it, because we're already simplifying it, the advantage of a crude model like this is that you can have very long run times. And so yes, it would be better to have water there, but no, we want to see what happens. And you'll see in a second why it is so important to have long run times. So now I'm going to show you a movie.
Because the origin of your rules could be the hydrophilic and the polar heads, right?
Absolutely. So you could make here, you could have water molecules with the water molecules having an attractive interaction energy with the blue groups and a repulsive energy with the yellow groups. Yes. But this aggregation is not caused by water. It is simply caused by the mutual attraction of the yellow groups. Is that okay? Good.
Yes, thank you. Yes, thank you.
Are there any other questions? No? So please ask questions. Okay. So here is the movie. So you see here 2 movies playing simultaneously, and I'm going to stop them at this point to explain what you're seeing. So the top movie shows the lipid bilayer over here. And the brown are these transmembrane proteins which are curvature generating. Notice that these ones are brown and this one is purple. That indicates that is because there was an additional switch which calls these proteins inactive and this one active. It happens when one of these inactive proteins meets with two or more neighbors. It indicates a conformational change. It so happens that very often there's a conformational change when capsid proteins come together. So these proteins do not attract each other. It's only when they form groups that it becomes attractive. So brown capsid proteins need to hit this interface here before it can become a purple one. So you got to seed it with a small purple cluster. So I didn't show that, but there was a small purple cluster for start. And from there on, it was growing. So initially it was growing pretty much like a flat disk, but because these are curvature generating proteins, it started to produce this curved shell. In this second picture, I don't show the brown ones and I don't show the interior. I only show the top. So this is roughly a half-formed shell, and it happened quite quickly. Already in a few moves did we see this form. So on the time scale of the simulation, this was a rapid process, and it is induced by surface diffusion of the capsid proteins to the edge of the growing capsid. Is this picture clear? These flies which you see flying around, those are lipid molecules which have evaporated because of thermal fluctuations. Is there any questions? Let me know, and then I'm going to play the movie a little bit longer. Whoops. Here you see it. So you see what's happening. The shell keeps growing. You should try to watch both pictures simultaneously. So we are nearly there. Look at it. It already forms a neck. Not quite. More proteins come in. They cause it to curve, the next form. And then look at it. We are only halfway through the movie. You might say we're done in a second. At this point, it begins to stall. So the growth does not continue. It stays here and stays here. And you run this molecular dynamics simulation for ages. This flash which you saw was because I cut out, or they cut out a large movie. And then suddenly it pinches off spontaneously. And here we have the capsid which has formed. This is now our toy viral capsid. I don't show the genome in here. So what
Excuse me, can I ask a question?
Please, yes.
Yeah. What are the equations of motion that you're using?
So the equations of motion are just Newton's laws for the interacting proteins with simple toy potentials, which I indicated before. I gave you this crude rules: yellow attracts yellow, blue repels blue, blue repels
They are
yellow.
non-realistic.
Yeah, they're completely unrealistic. But I'm thinking here as a physicist. I'm not interested in the microscopics. I'm not interested in the fine details, because my intuition is that this is a crude physical mechanism. It's not due to, why do I believe that? That's an important question. Again, a molecular biologist might say, oh, this is probably due to the intricacies of the interaction between the proteins. However, this phenomenon and this stalling is not specific to HIV. All enveloped viruses show stalling when they grow. There's always this problem. So that indicates that it's a universal problem, which is not a question of the specific interaction potentials between the capsid proteins and the interaction between the capsid proteins and the lipid. I already told you that for HIV, this is quite unrealistic, because there the capsid proteins are not transmembrane, they're just hanging below it. But I believe that this is a universal phenomenon. Whether that's justified has to be seen at the end of the day. Is that okay? So
I have a question.
Yes.
Yes, well, as you say, this is not really like the structure of the HIV,
No.
because the virus has a large area of bilipid layers, and
Yes,
the
it's larger.
proteins are quite separated. So they wouldn't form this conglomerate as this image.
Can you specify? I didn't quite follow. They are quite separated.
Yes, these proteins,
Yeah.
if they are in, simulating the receptor of the virus for T lymphocytes, they are quite separated along the surface.
Oh, okay. So you were thinking about, okay, you were thinking about these proteins on the surface, right?
Yes.
The recognition proteins. No, this is, okay. So first of all, this is a simulation which is more aimed at the alpha virus rather than HIV. Second, so yes, you are right. The surface, the proteins which stick out on the surface are quite separated. They recognize each other. And for HIV, the capsid proteins are attached to the inner membrane, but they also generate curvature when they start to cluster. If you have a cluster of GAG, they're called GAG proteins. So these structural proteins of HIV, when they stick together, they form curvature. Do you remember the very first picture I showed you, this thick shell of HIV hanging below the surface? That was intact GAG proteins, which had aggregated in an extended state, and that extended state forced curvature on the shell. So that is quite different from the alpha virus, and it is certainly not related to the surface proteins, which are for recognition. So I'm talking here about an effect which is generated by the GAG proteins below the surface. Okay. And I'm modeling that in a very crude way with these transmembrane proteins, because I'm after a physical effect which is quite independent. This kind of stalling you see for transmembrane capsid proteins, as well as for viruses which, like HIV, have capsid proteins which are membrane associated. That's the technical term. Do I answer your question, or?
Yes, thank you.
Okay. Robin, I have another question.
Please, Orlando.
Yeah. When this virus, molecular virus that you have here, is trying to go out from the surface, are not you losing the genus? Because the genus of the, because you are stopping to...
Yeah. You're saying, how about the Gauss curvature, right?
Yes, exactly.
Yes, of course. Yes, Orlando. So yes, if you imagine that this is part of the surface of the cell that has 4 pi, and then suddenly there's an additional 4 pi. So yes, you have a discontinuous change of the Gauss curvature. And that process is at the heart. So don't steal my thunder, Orlando. Yes.
But I wanted to point out that there are experiments with liposomes, bilayers, and adding up a polymer, and this budding or purling phenomenon is observed, but it never pinches off.
Oh, you're talking about like having a lipid cylinder and you tense it and you get purling on it and it doesn't. That's right. Absolutely.
That's correct. So I mean, just to add up to Rolando's question.
Yeah, but keep in mind that the purling phenomenon you're mentioning is for one component lipid system. The spontaneous budding happens for 2 component systems. So my guess would be, if you try to do the purling experiment, I think it was done in the Weizmann Institute, and try to do it for a 2 component system, which is on the border of phase separation, then that should, then you expect to get phase separation induced by the purling, and that might or might not induce formation of droplets.
Yeah, but your simulation is a bit misleading, because I mean, there's basically not 2 components, only one. So you arbitrarily say that one is inactive and the other is active, but
sort of like that, yes.
Yeah, okay.
But it would be fun to try to redo these old experiments, but now with multi-component lipids, which want to phase separate. Okay. Yeah. Any more questions? No, and then I continue. So in response to the question of Orlando, I immediately go to Carl Friedrich Gauss. Why does mathematics give me insight into this strange phenomenon? So once I'm going to go back to this Gauss curvature K. So here you see I want to have my nice laser pointer. Okay, here you see three surfaces. This is a sphere, this is a cylinder, and this is a hyperbolic surface. Okay, so when you define the Gauss curvature at a point, here the red point, you're looking at the maximal curvature, which is in this case along this direction. And we call the radius of curvature R1. And the minimal curvature, which is in this case perpendicular to that, it's actually always perpendicular. And it has a separate radius R2. And the Gauss curvature in this case is minus 1 over R1, R2, minus, because it's like for the lens formula of which you learn in elementary physics and optics, the curvature center here is outside, and this curvature center is inside. Now we go to the cylinder, then R2 has gone to infinity, R1 stays R, so that means that with this definition, the Gauss curvature is 0 for the cylinder, but notice it's still curved. That's described by the mean curvature. And finally, for the sphere, the Gauss curvature is positive. It's 1 over R squared, because the centers of curvature are all at the center of the sphere, of course. So the key point here, Gauss curvature is negative here, 0 there, and positive there. Go back to the Gauss-Bonnet theorem. I'm going to rephrase it as saying that the average Gauss-Bonnet theorem over the surface, if it's a closed surface, so you imagine that this is closed somehow and this is closed, is 4 pi divided by the area. So that's for a closed shell without holes. So this is the great Gauss in a nutshell, but that's all I need for my talk. So again, if there are questions, ask them. I'm going to apply that to this simulation. So the key point of the Gauss-Bonnet theorem is that the average Gauss curvature of a closed shell is conserved. So let's start with a flat sheet. Because it's flat, the Gauss curvature is 0. And now think of this flat sheet as the lipid bilayer we've been simulating. Since we have a conserved mean Gauss curvature, in the budding state, the mean Gauss curvature has to stay 0, no matter what you do. So let's look at the 3 parts of this. This is a picture of during the stalling. So in the Capsic part, the Gauss curvature is positive and is 1 over R squared. And this grows, so the bud area increases in time. The Gauss curvature here is negative, 0, and the Gauss curvature in the neck is negative. The neck area, you can see, decreases in area. It becomes smaller and smaller and smaller. But because the mean Gauss curvature has to be 0, it follows that the negative Gauss curvature of the neck must strongly increase in time as this neck narrows. Okay. That is simply a consequence of the Gauss-Bonnet theorem. If you have these curvature-inducing proteins over here, they unavoidably will create a neck. And that's why I want to hammer that it doesn't matter whether these are transmembrane proteins or membrane-associated proteins. If you bud off like this, you will form this neck with a negative Gauss curvature. So that is pure math. Here, once more, I emphasize it. Here, K is positive, the Gauss curvature. Here, K is negative. And of course, K is 0 elsewhere. Okay, so the negative Gauss curvature must increase in time. And now I'm going to introduce something from basic thermodynamics, the chemical potential, which is the Gibbs free energy per particle. 2 statements from fundamental thermodynamics. In equilibrium, the chemical potential is a constant and corresponds to a minimum of the Gibbs free energy. But we are obviously not in thermal equilibrium in the simulation. Maybe the final state is, yes, okay. But during a non-equilibrium state, when the chemical potential is non-uniform, you get currents. And these currents, according to non-equilibrium thermodynamics, are proportional to the gradients of the chemical potential. In our case, these are surface currents. And the surface current is a constant times minus the gradient of the chemical potential. That is completely general. There's nothing here about HIV. It is basic thermodynamics. So let's apply this idea to this structure over here. Okay, so first, the capsid proteins spontaneously assemble over here, which means in terms of fundamental thermodynamics that they have a low chemical potential. Okay, in other words, they like positive Gauss curvature. Next, if you looked carefully, you would see that while there is a certain density of capsid proteins here, which is moderate, and it's very high over here, the capsid, I forgot to point it out, but you can see it clear. The capsid protein density in the neck is very low. These toy proteins avoid the negative K region of the neck. In terms of thermodynamics, that would mean that the chemical potential in the neck is high. Finally, because the proteins spontaneously diffuse across the neck to come here, this must be intermediate. So the chemical potential here is higher than there. Otherwise, you would see the same or higher density. And the chemical potential here must be lower than there. So unavoidably, the chemical potential must depend on the Gauss curvature. That's for this simulation. And it's no surprise because these are curvature generating proteins, but they are coupled not only to the mean curvature, but also to the Gauss curvature. The simplest version you can do is a linear dependence. And since positive Gauss curvature lowers the chemical potential, the proportionality constant must be negative. So A and B are positive constants. Okay, so that's what I deduce from a thermodynamic analysis of the picture. Next, there is surface diffusion across the curved neck, but it has to go over an energy barrier because you don't see many particles here. Since here the curvature energy, the Gauss curvature is negative, that means that the chemical potential is high, which we already concluded. So in terms of the chemical potential is intermediate here, high here, and low there, which means that you have a kind of geometry-induced energy barrier. So that is really fascinating for a theoretical physicist. Why? Because the growth of the capsid alters space. It alters the surface geometry. From the mere fact that you're growing a capsid here, you necessarily induce a change in the Gauss curvature in the neck and thereby increase the energy barrier. So for a physicist, you can treat it as a toy black hole. As more matter goes into here, it creates more of a barrier to get there. Okay, I don't think that's a really good, but anyway, I like it. So the strange thing is here we have a surface diffusion process which is altering the space in which it takes place. I am not, well, one moment. I need to get rid of the pen. And so once again, here's the simulation. You saw now the seed. This is the result of a calculation using equilibrium thermodynamics. It's this red curve. So initially, let me stop this. Oh, what do I do? Okay, yeah, it stopped. So I'm not very smart. Okay, I want to stop it at this point. Good. Initially, I'm plotting here the current I as a function of the fraction of the capsid. So as the fraction of the capsid grows, initially the current, the total number of diffusing proteins along the surface reaching it, grows. Well, it grows because the parameter gets larger. That's what it does in ordinary 2D diffusion. So it grows. So the current seems initially to go faster and faster, solving these equilibrium. So what I did is our group solved the thermodynamic equation on a deformable space. The surface diffusion alters its own geometry. So you can do that. I don't want to bore you with the technical details, and this is the outcome. At this point, the current starts to decrease. Well, the reason it decreases is because Gauss curvature begins to form. And the Gauss curvature becomes larger and larger around the neck, and the current starts to decrease and decrease and decrease and decrease. And when about 0.8 of the neck has formed, it is practically zero. Again, because we've created a region of large negative Gauss curvature unavoidably because of the Gauss-Bonnet theorem, which the proteins can't pass. So the growth process inherently is self-suffocating. It suffocates itself because the positive Gauss curvature over here, which you want, necessarily imposes a negative Gauss curvature, which you don't want. Here are the data, if you can call them data, which come from our simulation. So the simulation, it's crude. It doesn't go to zero. There is pinch-off. This pinch-off is something which is not described by the continuum theory, and it's some molecular event where you rearrange the lipid bilayer so that it breaks into two parts. That is not described by the continuum theory. Predicts it shouldn't happen because as the Gauss curvature gets bigger and bigger, it just stops. In actuality, it doesn't stop. So in conclusion of the problem of the stalled bud, stalling is an intrinsic feature of budding driven by curvature generating proteins. It doesn't matter what kind of proteins is. It is due to basic mathematics and basic physics. They must stall. There's no choice. Second conclusion, the viral budding is fundamentally different from the Lipovsky mechanism. The Gauss-Bonnet theorem generates a geometrical trap or barrier, and eventually proteins can barely enter or leave. So does it help us in thinking about therapy? Well, a physicist would say, let's get rid of the escort machinery. That's fantastic. Not only would you get rid of HIV infection, all budding of membrane-developed viruses would be inhibited. Would be great. Unfortunately, suppression of escort, this is this pinching thing. It does happen. It happens because of genetic defects, and the results are not good. It produces neurological disorder. So we have learned new physics, I claim. Unfortunately, this insight won't help you come up with new therapies. Now, at this point, I would continue on with the problem of the second shell, but I am actually over my time. So I need to ask the chair what you want me to do. The second part of my talk would be another half hour, and I'm happy to discuss it with all of you privately, but this is a big audience, and the chair needs to tell me if you'd like me to continue on or not. Cecilia? Hello? Can you hear me?
Yep, we can hear you.
Okay, so screen sharing. So Orlando, what should I be doing? Should I continue with the talk or shall I just finish up here?
For me, there is no problem. I don't know everybody, but for me, it will be okay. It will be okay.
So can you ask the chair what would you like me to do?
Yeah, if the answer of a chair, I would suggest that people who feel satisfied could disconnect now, but we others continue.
That sounds good.
Yes, it's good. It's good. Good solution. It's a good solution.
Good. So then why doesn't the people who have attended and they are happy with this part, feel free to leave and I continue on. And when you say you've had enough of me, please, I understand. Okay, so I hope to go back to PowerPoint, my PowerPoint presentation. I need to go to slideshow, play from current slide. Good. Okay, so I have to turn my laser pointer back on. So now we're going to go to our next problem. And that problem I already announced in the beginning. Here you see a much more sophisticated image. It's a 3D reconstruction by cryo-EM by a leading person in the area, Briggs. I believe he lives in England, where you can see a far more detailed picture of the mature virus. This is the outside. the thin shell with the lipid bilayer and some remnants of the capsid protein. And here you see the second shell in red, and the medium in between is filled with stuff. Hello? Was there a question?
I have a question.
Yeah.
Why is the use of classical mechanics justified?
Ah, okay, thank you. In fact, my experience after I gave a talk in UCLA is, why is quantum mechanics not used? That's what you're asking, right?
Yes, exactly.
Absolutely a good question. Because 90% of my colleagues earn their living with quantum mechanics. So why is classical mechanics justified? That is a very delicate question. Basically, we are in a situation where we are at room temperature in a dissipative environment. So to see quantum mechanical effects due to interference and entanglement, you need to have a coherent wave function. Now, inelastic interaction with an environment, because of, let's say, the excitations of the water molecules, destroy the quantum coherence and will have a tendency to produce effectively classical results. That is, in a very short nutshell, the answer, which leaves unsettled the question that there are, in fact, subtle quantum mechanical effects in molecular biology, which you can see, but it is very delicate. So grossly speaking, when we do molecular dynamics simulation, even all atoms molecular dynamics simulation, there are special packages like GROMACS and CHARM. What people do is that they use potentials of interaction between the particles which come from quantum chemistry. So the interaction potentials themselves are fully quantum mechanical. Van der Waals interaction is quantum mechanical, hydrogen bonding is quantum mechanical, all of that. And that enters into potentials which then are put into a classical description involving solvent molecules where you do not take quantum mechanics into account. The interplay when you go to the single protein level, quantum mechanics starts to appear, and a method was developed by a famous simulator called Parinello in Italy, where you can kind of do both, where you can have a restricted quantum mechanical effect in a more classical protein which envelops it. Have I answered your question?
More or less, yes.
Okay, so feel free to ask it later. There are people with fantastic ideas about quantum mechanics and consciousness and all of that, but I'm Dutch, I'm a Dutch farmer, and that is way over my head. So we're just do it simple. Okay, all right, let's go on, unless there are more questions. So back to this picture. So these are reconstructed from multiple tomographic slides. So you take the same virus, you take different tomographic slides. The yellow here is stuff which comes from the host cell, all kind of macromolecules like RNA and proteins from the host cell, typically. There is not, in general, there are also remnants of probably of the maturation process. So why do we have a second shell? Notice, by the way, it is kind of starts and ends at the surface of the first shell. So here are some older pictures of the second shell in isolation. You can do that. And although it is not very clear, people deduce from these old pictures that these are closed shells of protein hexamers. Okay, and currently we can image these hexamers. Now, there's a theorem by Euler. And you know this in terms of the number of if you take a closed lattice composed of vertices and of faces and of edges, then there is a topological invariant. It's actually related a bit to the Gauss-Bonnet theorem. Okay, if you have hexamers and you close a shell and you apply Euler's theorem to that closed shell, then it follows that there must be 12 pentamers. And the interpretation of these shells, which you see over the different shapes, has to do with the distribution of these 12 pentamers. if you take 12 pentamers and you uniformly distribute them, you get a spherical structure. And if you put an excess of pentamers on one side, on one side and a deficit on the other side, and you get this typical ice cream cone shell. So there must be 12 pentamers, and modern reconstructions have seen the 12 pentamers. That by itself shows you that fundamental physics has something to, and mathematics has something to contribute. More recently, we have been able to image the RNA inside. So there are these 2 RNA molecules of HIV, which are inside, 2 trapped RNA genome molecules. Each of them are about 20,000 nucleotides long. So just to give you an idea, one gene, means one protein, is of the order of 3,000 RNA nucleotides. So if you have 20,000, that means you have of the order of 7 proteins. The longer the RNA, the more complex the program becomes. So there are 2 of them. It's interesting to ask why 2, why not 1? But for us, the analysis of these tomographic images allows you to kind of reconstruct the RNA. The RNA is not sticking to the surface. Here are the reconstructions. This is still from the group of Briggs. Here the RNA is sort of a dense blob at the edge. And in this spheral cylinder, it's sitting more in the middle over here. So most of the time, the RNA is kind of over here. I'll tell you later what this red thing is. This Are there questions? Okay.
Mira, está en un video, pero sí va a contestar.
Hello? I think you should mute, somebody should mute because I can hear very, okay. Thank you. So let's continue. We're now going to talk about this RNA, and we're going to do a little bit molecular biology. What is really strange about this second capsid is that it's permeable. Typically, capsids are not permeable for large molecules. You want to protect it against digestive proteins. This one has these holes here which allows small and medium-sized molecules to go through. So that is very strange for a viral capsid. So that makes the riddle even more interesting. So here is what's called the fundamental dogma of molecular biology. So here is DNA, double-stranded, and I'm sure you know about replication of DNA. There are proteins which can turn one molecule into another molecule. If you want to make a gene, you need to take a section of the DNA and copy it. And this is the copy. It's called this. You transcribe it and you copy it into a single-stranded RNA molecule. And then another cell machinery takes a single-stranded RNA molecule, translates the sequence of bases here, and produces a protein. So it is a flow going entirely from the top to the bottom. Double-stranded DNA, transcription, single-stranded RNA, translation gives you a protein. Now let's go to the kind of viruses like HIV. 2 people, Baltimore and Temin, got a Nobel Prize because they said that the basic dogma doesn't work for retroviruses. So retroviruses have, the word retro has nothing to do with cars or clothes. It has to do with reverse transcription. You go from RNA to DNA. People for a long time refused to believe it, but the evidence piled up, hence the Nobel Prize. So you go from single-stranded RNA to double-stranded DNA. That never happens in your own cells unless they are infected by a retrovirus or some other virus. Normal cell flow, the information goes strictly, you need them along a single direction. Okay, so something is doing this reverse transcription. It takes something for HIV is taking RNA and turns it to double-stranded DNA. So again, single-stranded RNA, double-stranded DNA. So let me just remind you a little bit about RNA and DNA. They are strings of elementary building blocks called nucleotides. Here is a nucleotide. This is a base, and the 4 different bases of Crick and Watson, that's the genetic code. It is attached to a charged phosphate group, and you link them together to form a chain. So you can either have a single strand, like an RNA. So this is the sugar phosphate backbone. This is a phosphate, that's a sugar group. So you have a charged backbone, and then you have these strands sticking out. That is this part. So that's single-stranded RNA. And here you have double-stranded DNA, where you get Crick-Watson base pairing CGAT. And I'm going to talk a little bit about the mechanics of the difference between RNA and DNA. But an important point is that they are both highly negatively charged. That will play a big role. This is necessary to keep them apart in the cell. Inside the cell, they are highly negatively charged molecules don't aggregate. Van der Waals attraction would tend to glob everything together, but this large negative charge prevents it. But you have the Watson-Crick complementary pairing. And the Watson-Crick complementary pairing producing double-stranded DNA produces relatively rigid molecules. Double-stranded DNA doesn't bend very easily. Thermal fluctuation will cause it to bend a bit on length scales bigger than 500 angstroms. But that's not together. It's true for RNA. For RNA, you can get self-pairing. So you can have Crick-Watson pairing between two groups, and you can form a kind of loop when you do that. So single-stranded RNA has a very complex geometry, and there are many configurations with comparable energy. So this is due to Crick-Watson pairing forming loops. So you can get complex tree-forming structures. So you call this the secondary structure. So there's a big mechanical difference between RNA and its secondary structure and double-stranded DNA. By the way, double-stranded RNA is also rigid. But in the context of today's talk, it's just DNA double-stranded versus RNA single-stranded. Okay. Now, who is causing this transformation from single-stranded RNA to double-stranded DNA? It is an enzyme called reverse transcriptase. And this reverse transcriptase transforms single-stranded RNA into double-stranded DNA if it is provided by DNA nucleotides. So here you see a cartoon. This is the enzyme, reverse transcriptase. This is single-stranded RNA, and it is moving along it. It's a motor protein. It consumes free energy. So it walks along the RNA, and it leaves double-stranded DNA behind. You need feedstock for that. You need DNA nucleotides, which can then feed in and produce this double-stranded DNA. Well, the single-strand RNA is just thrown away at the end of the day. So this is a motor which is also doing polymerization of double-stranded DNA, respecting the code of the single-stranded RNA. Is this sort of clear? I had a lot of time. I needed to wrap my head around how this works. But this is the heart of the retroviruses. So again, reverse transcriptase polymerizes double-stranded DNA from a single-stranded RNA template, and it's a motor protein. It can exert some force. It has to walk along the RNA. Back to the picture. This is the picture which you saw from the big groups. There's a blob of RNA, and here is the mature capsid. And I mentioned to you that there are holes here, and the holes are big enough to let these DNA nucleotides come through, so you can already start the transcription inside the capsid, inside this mature capsid, provided you have reverse transcriptase. Now, if you recall, you saw on the pictures of Briggs a red dot. That red dot is exactly reverse transcriptase. They apparently could image it. It is co-packaged. So this is actually a chemical reactor. It has single-stranded RNA, which is the substrate for the polymerization. It has the enzyme. And if it is placed in a solution of DNA nucleotides, the DNA nucleotides can diffuse through. Reverse transcriptase can start its work and turn this very flexible RNA molecule into a much more rigid DNA molecule. Are there any questions about that?
No.
No? on we go. Why? Okay, again, this is when I ask a molecular biologist why, he just looks at me as if I've lost my mind. Evolved like that, and that's that. Okay, so you remember that I like Sherlock Holmes, and if you are a Sherlock Holmes aficionado like me, you know about Professor Moriarty. Professor Moriarty is the great opponent of Sherlock Holmes, and he is pure evil. So this transcription of RNA into DNA looks to me like something satanical practically. So let's go back to this picture. Here is assembly, budding, immature virus, mature virus. So now we follow the mature virus, zip, 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 zip. And here are these surface receptors, which were mentioned by one of you. They recognize a molecule on the surface of the cell called CD4. That causes fusion by itself, very interesting. Fusion of the membrane of the mature virus with the host cell. Here is the leftover bit of the membrane. And now the mature capsid is inside. Once it is inside there, it can start, it uncoats, that is the name, not a fancy name for just it falls apart in some fashion. Then reverse transcription takes place, and here you have double-stranded DNA viral material. So the reverse transcription is not shown here, it just takes place over here. And here is the trick. This DNA is imported in the host cell and integrated into the host chromosome. Then, when the host cell reproduces, it just copies the double-stranded DNA from the, which originally came from the virus. And at some point in time, it will trigger transcription of this genetic material, and you start to make RNA genomic material and protein material. So you get viral mRNA, viral proteins, and it self-assembles and it starts all over again. What many of my physicist colleagues don't realize is that they carry tons of this viral RNA with them. A lot of our, something like 10% of our genome is nothing else but a reverse retroviral material, which decided to stay with us. It doesn't get transcribed necessarily, it just lives with us. It piggybacks on our reproduction. When you reproduce also lots of viruses. And strangely, they are able to move around on the genome, and we call them transposons. And there's a whole interesting story about them. So at any rate, the retrovirus injects its own genome. Sometimes it can cause cancer, luckily not for humans, but there are animals which get cancer because this integration process disturbs the chromosome and produces cancer. The Rous sarcoma virus is sort of an example of that. So here you see the satanic organization of the retrovirus. What I'm going to talk about is this part of reverse transcription, because my coworker, Julia Rozina, demonstrated to me that this is very unlikely. This is a standard picture, and we don't believe it. Let's tell you why. So the retroviral DNA is inserted into the genome of the infected cell. I mentioned it can cause cancer, and now we can go back to the original question about the second shell. Why is there a second shell? So let's look a little bit at the mechanics. So as long as the RNA, as long as the genome is an RNA form, it's a blob, it's a deformable blob. You remember it has these arms, it's sort of like a branched polymer. It can easily be compacted. It's not stiff at all. It easily fits in the second shell. It only fills up a little bit of it. However, if you take that roughly the same length of RNA and transform it into DNA, which is much more rigid, I've tried to make this to scale. This is an atomic force microscope picture of the double strand. It's not of HIV, but of something of comparable length. And here I've tried to draw to scale the mature capsid. This is not going to fit very well into there unless you provide a lot of force. And you can convince yourself that if you provide enough force to compactify it and put it in here and then let go of the force, it'll burst it. It'll burst this mature shell. So swollen, soluble Hiv-1 DNA will not fit in here. So that is really strange. Now, for a physicist, that rings the bell. Soft matter physicists have known for a long time that, in fact, there's a second version of double-stranded DNA where it is compact. And that's called DNA condensation. So you can take viral or bacterial DNA and you can compactify it very easily, make it really much smaller. What you need to do is add condensing agents, positive polyvalent ions. So if you start with a highly swollen large DNA molecule and you add ions, positive ions with a charge of +4, it will beautifully roll itself into a torus. Here is a top view, here is a side view. I don't know what is the difference between this and this. This is a classical picture. It has been stained for you. If you look at the cross section over here, it has secondly ordered. So it's like a bun. By the way, you see these facets here. It's really like a two-dimensional crystal, which is wrapped around into a torus. This crystal is rigid. You can't really make it any smaller. The positive polyvalent ions can thereby greatly reduce the size. So I want to show you this picture of purely naturalistic reasons. So here is the double-stranded DNA. And here is a particular polyvalent ion called spermine. Spermine has a charge of +4. So here you have a lot of negative charge and here you have compensating positive charge. The spermine compensates or charge neutralizes the DNA. It no longer repels and you can form crystals. In fact, it's a little bit like a salt crystal. And the fancy word for it is a Wigner crystal. This is not the Wigner crystal of electrons, which the condensed matter physicists among you are very familiar with. This is a Wigner crystal of ions. We have just stolen the name. I've heard no complaints. So we have here a crystal of negatively charged DNA and large positively charged ions. And I am proud to say that this particular ion, spermine, has a very simple structure, was discovered in the Netherlands in 1677 by Antony van Leeuwenhoek, the great microscopist, in fact, the first microscopist. He discovered spermine in male sperm. All the males in the audience have sperm cells where there's about 1 meter of DNA condensed in a micron-sized package, and it is condensed through spermine. And of course, Van Leeuwenhoek didn't know the chemical formula, but he was able to isolate the spermine. I think if you look at this date, 1677, that is a fantastic scientific accomplishment. Okay, so is Hiv-1 DNA condensed in this way? Is there spermine hanging around in HIV? No, there is no spermine. So I want to remind you of this process at the beginning of the talk where you take this large GAG protein, the structural protein. This is the membrane associated part, MA. This is CA, the capsid associated part, from which you make the mature capsid. And here's this little bit hanging around. This little bit is called NC, nucleocapsid, and it is positively charged. And in fact, it is this positive charge which causes the RNA to adhere to the inside of the immature capsid. During the maturation, you cut this link. So what happens is that for every digested GAG protein, you produce 1 positively charged peptide. It has a charge of 3.5, just a bit less than spermine. So when you have your immature capsid, you have of the order of 1,000 or 2,000 of proteins. It varies actually. And during the maturation process, you generate that number of about 2,000 of these NC polyvalent ions with charge of +3.5. And it is enough to, if you do the arithmetic to charge neutralize the RNA. So you generate maybe about 4,000 polyvalent ions, and it's enough for charge neutralization. HIV generates its own polyvalent ions for condensation. Okay, all right. So now we think the problem is solved. We have learned that we can condense the viral DNA into a tight little package. Does it fit? It is just about the same size. In fact, the torus is still a bit bigger than the shell. The diameter of the double-stranded DNA is somewhat larger than the maximum diameter of the mature capsid. That's a little strange, right? And it cannot be a coincidence. Okay, you need an experiment. And now I'm actually going to enter on this key question of physicists interacting with life scientists. A beautiful experiment was done by a physicist, Itai Russo, in the Weizmann Institute. And I'll tell you a story about Itai. He did what I think a brilliant experiment. He took the mature capsid with the RNA inside and isolated them and put it in a solution. He then studied it with an AFM and scanning electron microscope so he could measure the tension, okay? So initially, it is all RNA. So here is a stiff shell, which has RNA inside, and it is pretty stiff. He pokes it with an atomic force microscope and can measure the stiffness. What he then does is he flows in the nucleotides, the DNA nucleotides. It's the reverse transcriptase is packaged in here. The nucleotides can diffuse in, reverse transcriptase can start, and it can synthesize more and more and more DNA. The torus grows, and guess what? It ruptures the mature shell. It ruptures it when it is nearly complete. So here's an example, here's an example. Sometimes you see two ruptures. The tension collapses. So when you go from here to here, suddenly you lose when the integrity of the shell has been compromised, you lose the tension. And in fact, the DNA can go out. You lose the DNA. So then you say, aha, now everything becomes clear. We have here a kind of chemical reactor which is leaky enough to allow the foodstuff to come in. It protects the enclosed genome from hostile digestive enzymes. And it breaks open, it's like a form of online delivery. It breaks open just at the right time. When it is finished, the DNA can come out. This is probably happening in the cell on the surface of nuclear pores. So this is, to me, was an answer to a big riddle. Now, here comes the sad story. Itai was coming up for tenure in the Weizmann Institute. And so the physicists who have no understanding why this was a brilliant experiment, do you know what they did? They asked life scientists to evaluate it. The life scientists were laughing. Who cares about AFM? This is not a natural environment. And so they gave a negative evaluation. So you see here the cultural conflict of working in an interdisciplinary area where the two sister disciplines, physics and molecular biology, have a different language in which they don't understand each other. And a person like Itai, who does a beautiful experiment, doesn't get appreciated, tragically, I think. So let's talk about some basic physics. Who's doing the work for breaking the shell? It must be reverse transcriptase, and it's a motor protein. It consumes the free energy, which is obtained by the polymerization of the DNA nucleotides. But it doesn't directly apply a force on the capsid. The force is exerted indirectly by transforming the flexible RNA into a rigid torus, which grows in size, one nucleotide at a time. So it's a little bit like an Archimedean lever when this thing grows. The force you apply on the shell is indirect. This protein is not forcing the shell. It's just walking around, and this thing grows. So it's a little bit like a lever. It applies practically no force, and by by sewing together the double-stranded DNA, it applies a significant 10 piconewton level force on the outer shell and causes it to break. So I feel that this is the answer of the question, where does the second shell, why do you need it? The second shell is a leaky, fragile reactor vessel. It prevents loss of the precious transcriptase. If this would float away, if there was a hole where it could allow it to escape, then everything would be lost. This is our crown jewels, but it does allow entry of the DNA nucleotides. It spontaneously fractures when the reverse transcription is nearly complete, and it protects the viral RNA from enzymatic digestion by host RNA digesting enzymes. So you cannot learn about it purely from a physics, purely from a molecular biology viewpoint. You have shared viewpoints which begin to give you insight. This is a few slides about whether you can apply this idea to look at therapy, but I think I should finish, and I want to thank my collaborators. First of all, the key ideas of the second part of my talk were from Julia Rusina, my collaborator. In the first part of the talk, the key simulation was done by Michael Hagen. I mentioned to you that I come a lot to Mexico City and I visit UAM, and these are my collaborators on a completely different subject involving HIV, and we are doing that together with Julia. And I'm sure that either Paola or Orlando would be delighted to come and tell you about it. And these are the students who worked on it. Sanjay Dharmavaram, who's now in Bucknell University, Celine Bao-Shen-Shi, she's an undergraduate now in Stanford, and my colleague, Joseph Rudnick, who is my friend and who is just retired from being the Dean in Physics. And I want to thank you for all of the time you have given me. I appreciate it. And that's it. So maybe I should stop share. So, oh, I have to turn off my automatic stop share. Okay, Orlando, good to see you again.
Thank you very much for your talk. I don't know if someone have some questions.
I have a question. No, I am confused because it seems that you are implying that when the virus infects T-CD4 cells, it injects the capsid, where the RNA is
yeah, the mature capsid, yes.
The capsid, but you don't really see that in the experiments, no? You don't see images of cells with the capsids inside, no?
Okay, let me do share screen, because the thing which I skipped, go back to here, discussed an experiment. So here is an experiment by Arhel from 2009. The question you asked is, can you see the mature capsid inside the hosts, inside the cytoplasm of the host cell? Was that your question?
Yes.
Okay. Well, it shouldn't be there, because once this is injected, the reverse transcription process starts, and you're destroying the capsid. So the only way to see the mature capsid, which is indeed not easily visualized, I do not know if it has been seen. If you wanted to catch it, you'd have to catch it immediately after entry, or you have to make sure that there are no DNA nucleotides in the cytoplasm. That would be my explanation. So the evidence is indirectly. To test for this description, what you can do is you can inhibit reverse transcription. There are various drugs which can do that. And when you do inhibit reverse transcription, you get what's called hyperstable core mutants, and then you start to see the capsid survive. On the other hand, you can also use mutants where you change the stability of the core, so the stability of the capsid. If you increase the stability, you again start to see capsid. If you decrease it, you see nothing. But here is the interesting thing. Whether you increase or decrease the stiffness of the shell, in both cases, you inhibit infection. So the stiffness of the shell seems to be delicately balanced so that it opens up at the right time, probably when the mature capsid docks in the nuclear pore and causes some additional stress. My collaborator, Julia Rusina, I don't know if she's attending, she will know better about experiments searching and detecting the mature capsid. So I am not sufficiently cognizant of the life science literature about to what extent people have been able to detect it. My guess would be it is difficult to detect them. If Julia is in the audience, then she can just wave her hand and tell us about it. So I do stop share.
Okay.
I have a related question.
Okay.
For the pinching of the first membrane,
yeah,
which you mentioned that is aided by a sort of molecule that makes a ring,
yeah,
and favors the Gaussian thing and all that.
Yeah, it seems to detect Gauss curvature, and it sort of attaches itself to that. It forms a ring, and then it is activated, and it's like it contracts. Yeah, go ahead.
We are doing several experiments in bacteria preventing mitosis of.
Yes, of course. So the way, and it's very similar to the way a bacterium divides by contracting a ring. But those are actin filaments, I believe.
Yes,
I mean, not
ring. But we actually predicting theoretically that mitosis could be inhibited if you change the elastic modulus of the membrane.
Mm-hmm.
And that you could do by adding up some hydrophobic molecules that penetrate the membrane and increase the modulus, the bending modulus of the membrane. I wonder if somebody has suggested.
No, but I don't know how strong your membrane has to be to stop the process. So you're telling me that if you stiffen the membrane or strengthen the membrane, you can stop the mitosis. Exactly. So the scale is a little different. It's on a larger scale. Your system has a little bit larger size.
Oh, yes.
Maybe not, maybe not. So no, I do not know experiments. It sounds like a nice idea to strengthen and see if you can thereby inhibit viral growth, because so it has to, then you need to know the mechanical strength or the force which the escort machinery generates. And I believe
easier because of it's a smaller scale, so you would need less modifications of the membrane.
I think it's a very nice idea.
Well, thank you. So we will pursue it.
No, one thing you could imagine doing is finding small molecules which also try to sit on negative Gauss curvature region, but strengthen it rather than weakening it. But it seems to me that is something which needs to be addressed by molecular biologists who understand these things better than I do. But I think it's a nice idea.
We're using caffeine and nicotine, which...
Really? Okay.
Is there any more questions?
Yes, you said that the viral DNA is not integrated into the cell DNA.
Yeah, it is. It is. The viral DNA is integrated into the cell DNA. Yes. Sorry.
Oh, okay.
That's the key point of a retrovirus. The viral DNA, I don't know how to do that quickly. The viral DNA gets spliced into the host cell. So it just sits there and it can be expressed at any time. So a person with HIV infected cells, these cells can hang around for quite a while until there is some signal which causes them to divide. And in that case, you start to, you activate the transcription. So the general idea is that, well, let me take one step backwards. So most viruses have an RNA copying machine, RNA polymerase. HIV lacks it. It is entirely relying on ordinary gene transcription, except that it inserts its own genes into the host cell and thereby gains, so to speak, the protection of the host cell. And maybe if the host cell gets stressed, that could be the signal for the transcription to start of the viral DNA. And then you start to make viral RNA, which then gets transcribed, which then gets, you then couple that to the host cellular machinery, produce proteins. So you get viral proteins as well as viral RNA.
Okay, Fernanda Gutierrez has a question.
Please.
Can you go?
Yeah.
Fernanda, please tell us your question.
Sorry, yes. So I'm Fernanda Gutierrez. I'm a physician, HIV physician.
Hi.
I would like to ask you why then the virus inside of the capsid has 2 RNA strains.
Yes. I think that is one of these unsolved puzzles. I love it that you ask it, because I asked my own, why shouldn't one be enough? An easy engineering answer is, well, by having 2, you can be a little bit more sure that you're not making a mistake. So it turns out that during the assembly of the virus, in the very first stages of the assembly, when one RNA molecule associates with some gag proteins and attaches to the plasma membrane of the host cell, it cannot produce, it doesn't grow, so to speak. When 2 of these nuclei, so you have a sort of a nucleus of gag proteins with a single RNA, when they come together, they fuse, and there's a little, I'm sure you know, a palindromic sequence. So at the end of the RNA, there's a palindromic sequence causing them to fuse. And that's the signal that the whole thing can start to grow. To me, it looks a little bit like a safety device. You don't want to start it if you're not totally sure that you have the right RNA. You may remember I mentioned that unlike most viruses, retroviruses don't have RNA polymerase. The result is there are relatively few viral RNA molecule in the host cell. And typically, maybe about 10% of the, or something of that order, 10% of an HIV virus contains host RNA, not viral RNA. So HIV has to work very hard to distinguish its RNA from host RNA. Packaging host RNA is totally non-productive. By having 2 molecules which have to recognize each other, you're trying to make yourself doubly sure that you're not packaging host messenger RNA. So and it's a little bit like a nucleation problem. For a single RNA, the nucleus is too small. By having 2 and then fusing together, the nucleus becomes big enough for the process to start. It's very hand-waving, but that's my view.
Thank you. It makes sense. Yes, of course.
Okay. Thank you very much to you, Robijn, to everybody to being here. It was a wonderful talk, so thank you very much.
And I hope to come visit you when I'm allowed to travel.
I hope you another time, and probably in another city.
Yes.
You have been here so many cities with us, so I hope that it will be in the near future. Please give my best regards to everybody in LA.
And I would like to thank Grisel for inviting me. So thank you very much. And for being very patient with me with getting everything organized. I really appreciate it.
Okay. Thank you very much to everybody. And to you, Robijn.
Till next time.
Okay, till next time.
Thank you, everybody. Bye-bye.
Thank you.